I am Cory Tobin, the co-founder and chief scientist of Mozza Foods. For the past 6 years, our company has been engineering soybeans to produce casein proteins for use in dairy applications. So these are recombinant milk and cheese that we've made on very small scales from soybeans. And you can see there the casein that comes from soybeans is the exact same protein that's found in cows, and it has a stretch you expect out of mozzarella. The reason why cheese and other dairy products have particular properties is because of this fundamental unit of protein inside of milk called the casein micelle, which is composed of alpha S1 casein, alpha S2, beta, and kappa. And alphas and beta are hydrophobic proteins that tend to aggregate in the core of the micelle, and the kappa casein is partially hydrophobic, partially hydrophilic, and forms like a shell around those hydrophobic proteins. And these proteins are all highly phosphorylated, which allows them to bind calcium phosphate. And so in the context of mammalian feeding their young, it's a good source, it's a good vehicle for delivering nitrogen, carbon, calcium, and phosphate to the neonates. In the context of the human food supply, this is responsible for most of the properties that you expect out of cheese and other dairy products. So the kappa casein is hydrophilic and helps keep the micelle separate and prevent it from aggregating into like one big mass inside the cell where it's produced. But when you add rennet during the cheese making process, you're essentially cleaving off the portion of the kappa casein, which allows the casein micelles to aggregate, and in the process, it binds all the fat globules and everything in there into the big gooey mess that we like to eat. And so we produce these casein micelles directly inside the soybean. So this is an electron micrograph of a casein micelle inside of a soybean. So we've taken a soybean and sliced it very thinly with a microtome, and then stained it with antibodies for, I think in this case, beta casein that has gold nanoparticles attached to the antibodies. And so the electron microscope is able to pick up the contrast with those gold nanoparticles, and we can see where the casein micelles are. So this is a protein storage vacuole surrounded by oil droplets, and the casein micelles are in the protein storage vacuole. And then here on the right, you can see, as I was telling you, kappa casein forms like a shell around the outside of the micelle. And in this case, we were very fortuitous that the microtome made a clean slice through the micelle, and this anti-kappa antibody is forming a nice little ring around the micelle. So we can make casein micelles inside of soybeans and purify them out and make little cheese balls out of it. Hopefully in the future, bigger cheese balls. We gotta feed the world. We do our research in this greenhouse, which is a converted cannabis grow operation, which now grows soybeans. And we grow outdoors in Wahpeton, North Dakota, just south of Fargo. And I say this because all of our research is being done in this vertical greenhouse, which is very expensive to grow plants in. Very cheap to grow soybeans out in the field, very expensive to power the lights and the air conditioners and everything for the greenhouse. And so we're constantly trying to avoid putting plants into the greenhouse and having to maintain lines and screen plants in there. Soybeans are much bigger than Arabidopsis or other plants that people like to work with. Take a lot of space, a lot of water, fertilizer, light. And so throughout an entire screening process, we're always trying to minimize how many plants we put in here. We can make casein micelles inside soybeans. What are we doing now? We're trying to increase the protein expression levels in the plant to get to the point where we're matching cost parity with cow milk. And so our workflow is generally design plasmids, build the plasmids, transfer the plasmids to agrobacteria, which is the bacteria that can infect the plant and deliver our genes into it, transform the soybeans, screen the plants by genotyping, insert mapping, and then do all of our functional analysis on the RNA and protein to figure out which lines are the best, and then learn something from that and do it all over again by designing new constructs and doing the whole process again. And so we're always trying to minimize the time it takes to go from like every single loop of this because plants take a long time to work with, and so we're trying to shrink the whole thing down. The more of these cycles we can do in a year, the more science we can get done before we run out of money. So try to make this as fast as possible. So our workflow is pretty typical of people doing synthetic biology or molecular biology. design plasmids in SnapGene, generally 20 to 30 kilobases with up to like 12 coding sequences and regulatory RNAs. We then have some software that automatically takes a SnapGene file and generates protocols for the lab techs to build the plasmids using Golden Gate or MoFlow. We then pick a couple colonies off the plate, mini prep it, drop it in the drop box. And so I think it's pretty typical. I mean, we've done a lot of work in like optimizing the workflow because we're building a couple thousand plasmids every year. So it takes a lot of small wins here and there to like figure out how many things you can assemble at once or whatever, but pretty standard workflow. But one thing I want to point out that Plasmosaurus has allowed us to do is, back in the dark ages, as Jim was pointing out, we would have to pick like a lot of clones at the same time because the QC process was either like primer walking, which took a long time, or there's a couple services that would do like Illumina short reads on plasmids, and it was like a 2-week turnaround time. And like, I don't got 2 weeks, so I got to pick like multiple clones to hopefully get one good one in there, because I can't afford to like send one clone, wait 2 weeks, get the results back, send another clone. Like, it takes way too long. So now what we do is we pick 2 colonies off the plate. One of the liquid cultures we just set aside, leave on the bench for the day. The first clone 1 we mini prep and leave a little bit of liquid culture on the bench for the rest of the day. Drop off the mini prep in the drop box, get the results by 11 p.m. We have an automated system that grabs sequencing results off the website and compares it to our SnapGene files, and then instructs the night crew to make glycerol stocks or make labels for all the tubes in the middle of the night. So when I get in the morning, the only things that have been put in the archive in the freezer are things that pass QC with Plasmosaurus. And this is super important because with plant biology, like once I transform a plant with this stuff, if someone like accidentally grabbed the wrong glycerol stock from the freezer and transformed it, like I'm not going to find out until like 2026 or 2027, like later on, like way down the line, and be like, oh man, I wonder if we picked the wrong glycerol stock from the freezer that one day. And so this enables us to, we never make any glycerol stocks or archive anything until we have QC, and so we just like leave stuff on the bench until we get the results later in the day, which is a huge game changer for us. And if the first one comes back as bad, we have a second liquid culture sitting on the bench, and the night shift just mini preps it right there and sends it off for Plasmosaurus. And on day 3, we have the second clone sequence as well. And so it's like really changed how we do plasmid construction. We then pick a couple colonies off. We transform into agrobacteria, pick a couple colonies, mini prep at this time because the agro has like a very low copy or copy number of the plasmid, so you got to grow a lot of bacteria to get enough to work with. Digest it, compare it to simulation in SnapGene, and then archive the good clones we have, and then transform it into soybeans. So we take a cotyledon off of a imbibe seed and transform with the agro, regenerate shoots and then roots, have a whole big plant here to do genotyping on. So the thing with agrobacteria is it inserts randomly into the genome, which is a problem. I've seen some talks here about targeted insertions in soybeans, which is super interesting to me. But right now, we're kind of stuck with you transform your plant and your construct integrates randomly in the genome, literally anywhere. And in any copy number, it could be 0, 1, 2, 3, 4, 5 copies of it. If you have multi-copy inserts, it's most likely to get silenced. So I don't want to deal with those. Because we've done thousands and thousands of transformation events and seen which ones produce a lot of protein, we have a good idea of where in the genome are good locations for our insert to land. And so like land the chromosome 12, like done, I don't want that one. Nothing expresses good on chromosome 12. I don't know why, don't want it. So we want to figure out where our inserts are landing and how many there are. And later on do like measure zygosity so we can generate homozygous lines. So we take a leaf punch, do qPCR in-house to do the copy number and zygosity measurements. But then we also needed to map the location where the insert landed in the genome. So we do this process called repeat anchored integration capture. Wrote that one down. This is a process to help tell us where the insert landed. So we take genomic DNA, chop it up with a restriction enzyme, do some end repair and dephosphorylation steps, and ligate linkers onto the fragments. And then we have a biotinylated probe that targets our construct sequence, which we can pull down with strep beads. And we do a couple rounds of pull downs with PCR with a primer that binds to the other adapter sequence. And then finally, that like the amplicon at the end of that, in theory, represents like the boundary region of where our insert, like part of our insert sequence, and then part of like the genomic context in the endogenous genome. So we send that amplicon off for Plasmosaurus, and you can see here it's kind of like mapping successfully to our construct, and then all of a sudden it stops mapping successfully. So we take this sequence right here, blast it against the soybean genome, and that location is where our insert has landed. And then I was saying we have a pretty good idea of good locations and bad locations in the genome. Not perfect, but like good enough. And so if it lands in some locations that we know are to be bad, we throw the plants out because, as I was saying earlier, I want as few plants in the greenhouse as possible to minimize cost. And so I'm always trying to like weed out stuff as fast as possible. So now we have soybean lines that have our genes in it. We know we have these single insert events that we assume to be decent inserts. We can do gene expression analysis on it. We just started doing this with Plasmosaurus. I mean, in the past, we would do it with other vendors, but it would take 2 months to get the results back. And so by the time we get the results back, that plant's already dead, and we've already harvested seeds from it. Because we want to, we're not guaranteed to have a good insert. We want to screen plants for which ones have good inserts. And so we can look for, are our genes highly expressed in these transgenic lines? And, high expression of casein genes doesn't necessarily guarantee that you're going to have a lot of protein expression. There's like degradation pathways that are at play there. And ultimately, I don't care. At the end of the day, I don't care about RNA. I can't eat RNA. You can, but it's not tasty. All I want is protein. But doing protein analysis is difficult and expensive. So we kind of use this as an early screen, but now with a rapid turnaround time. Like by the time I get the RNA-Seq results back, the plant's still alive. I can keep working on that plant, which is super cool. Another thing we can do is, as Justin was saying, we can kind of use it as a qPCR assay. So one thing that we're interested in is the phosphorylation state of casein proteins. As I was mentioning, it's important for the biophysical properties of the protein. Beta casein in particular, when it's phosphorylated, has a shift on SDS page shells. So we can kind of get an idea of our proteins being phosphorylated based on a Western blot. And so you can see here, sample A appears to have a lower band than everything else. And so it's presumably not phosphorylated for whatever reason. The RNA-Seq said we were expressing caseins really well. Why is this casein not being phosphorylated? So we co-express this kinase called FAM20 along with the caseins, which is the kinase responsible for the in vivo phosphorylation of caseins in cows. In this case, we'd already done RNA-Seq on these samples, so I went back and pulled out the CPM values for FAM20 gene that we had expressed in there, and saw that it's much more lowly expressed in sample A than it is in B through E. And so now I know. Now I kind of like believe this Western blot more, that these proteins are in fact dephosphorylated or unphosphorylated. And why? I don't know. Problem someone else, not me. Plant A goes in the trash. Don't want that one. And then at the end, we do protein analysis. We do a lot of ELISA screens, but as with any immunological assay, there's a lot of problems with that. The gold standard for doing like protein quantification is LCMS or isotope labeled mass spec or PSAC. So what we do is we generate like heavy isotope labeled casein proteins generated with cell-free synthesis. And we have like a really good idea of how much, like what the concentration of this heavy casein is in the sample. And we blend it with our protein extract from the soybean, load it on SDS page gel, cut out the band where we assume the casein to be, send that to our partners at UT Southwestern Proteomics Core, and they do a trypsin digest, which cuts after every arginine. The sample now has heavy casein in it and our regular casein, And we know exactly how much of the heavy casein is in there. Both the heavy and the light casein get chopped up equally with the trypsin. It goes through liquid chromatography and then goes through an orbit trap. And where you see the orbit trap, it basically gives you mass to charge ratios of the different peptides. And at this point, the heavy peptides have a shift in the spectrum. And so you can see like, okay, this spectrum is for alpha casein peptide 16 through 25 or whatever. And you integrate the curve for the heavy casein peptide, and you know exactly where the light casein peptide is in the spectrum. And so now you can do like a ratio of the integration of heavy to light, and that tells you how much of that peptide is in there based on knowing how much, exactly how much of the heavy peptide was loaded in there. And so in this case, this plant has between 6 and 7% of the total protein being casein protein. It's pretty good, not good enough. Casein's cheap, and so we got to get to like 20-ish% to be cost parity with cows. So we still have work to do. And that is the end of it.